So let's start. First of all, we'll talk about the type 1 hypersensitivity reactions. Well, in type 1 hypersensitivity reactions, an antigen from the environment stimulates the B cells for the production of IgE antibodies. These are specific for that antigen. Now, overproduction of these antibodies is actually followed by binding on the membrane receptors of the Fc part of IgE on the surface of mast cells. Do you remember which region of antibody is called the Fc region? Okay, let's recall the two parts of the antibody. So this is what an IgE antibody looks like. It has two fragments, the upper fragment, which is the Fab region, and the lower fragment, which is the Fc region. Now the AB in FAB stands for the antigen binding, because this is a fragment where antigen will bind. And the C in the Fc region stands for crystallizable, because it is a well-conserved amino acid sequence that allows this fragment to crystallize. And this is the one that is responsible for binding to the receptors and initiating a biological activity. Okay, these mast cells that are coated with IgE antibodies are actually said to be sensitized. So next time when our body is actually exposed to the same antigen, it gets cross-linked with the bounded IgE, and this leads to degranulation. That is the release of mediators like histamine, leukotriene, and prostaglandins. These mediators will act on different protein receptors like G protein coupled receptors that are located on the surrounding tissue. And what will they actually do? They mainly cause vasodilation and smooth muscle contraction. Remember that the common examples of type 1 hypersensitivity reactions are asthma, allergic rhinitis, and systemic anaphylaxis. Okay, so coming to type 2 hypersensitivity reactions. Now, these are cytotoxic or antibody mediated. You can remember this by as type 2 can replace the cytotoxic. And it consists of two components, the antigen and the antibody. So in type 2 hypersensitivity reactions, antigen on the cell surface combines with either IgE or IgM antibodies instead of IgE antibodies that were observed in type 1 hypersensitivity reactions. Now, the IgM antibodies actually exist in the form of a pentamer, as you can see here. Now, if we discuss the different regions of this antibody, you can remember that this was the Fab or FAB region, and this is the Fc region. Now, this antibody actually attaches to the antigen via its Fab region and acts as a bridge to complement via its Fc region. Now, this antigen-antibody reaction activates the complement. Now, due to this activation, different phagocytes are attracted, and enzymes are released that damage the cell membrane of the target cell. If we talk about the examples of type 2 hypersensitivity reactions, we have hemolytic anemia, ABO transfusion reactions, or Rh hemolytic disease. And some drug allergies may also be categorized under type 2 hypersensitivity reactions. Like penicillin allergy is quite common in few people. Now, the examples of type 3 hypersensitivity reactions are serum sickness syndrome and Arthus reactions. Okay, let's start with type 3 hypersensitivity reactions. Well, type 3 means three things stuck together. These are the antigen, antibody, and the complement. In type 3 hypersensitivity reactions, again, IgG antibodies bind to circulating antigens to form an antigen-antibody complex. Now, this complex then deposits in particular tissues, and these tissues are mostly blood vessels. Now, this deposition activates the complement cascades and attracts the neutrophils. Now, the question arises, what do these neutrophils do? Well, these neutrophils will release lysosomal enzymes and cause cell death and inflammation at this site. Now, coming to type 4 hypersensitivity reactions. These are also termed as delayed hypersensitivity reactions, since they are a bit different from the first 3 types of hypersensitivity. Let me tell you that type 4 is not a function of any type of antibody, like the other types of hypersensitivity. Rather, it is a function of T lymphocytes. So we can say that it's a cell-mediated reaction rather than antibody-mediated reaction. Well, the response in type 4 hypersensitivity reaction is a bit delayed, meaning that is not initiated right after the contact with the antigen, but it takes some time and starts within hours or days after the contact. This is because specific T cells must migrate to the site where the antigen is present, and this process takes more time than reactions involving antibodies. So this transfer is not by serum, rather it is by immunologically committed, sensitized T cells. So on the basis of subtypes of T cells, types of antigens, and effector mechanism, type 4 hypersensitivity reactions are further categorized into 3 subcategories. So these 3 subcategories are CD4 T helper type 1 mediated reactions, CD4 T helper type 2 mediated reactions, and lastly, CD8 cytotoxic T lymphocyte mediated reactions. Type 4 reaction starts when the antigen enters our body. Now, this antigen is taken up by Langerhans cells. These are tissue-resident skin macrophages, and they act like antigen-presenting cells here. These cells then migrate to lymph nodes and form sensitized T lymphocytes. So this is the first step in type 4 reactions, that is the sensitization of T cells. Now, these pre-sensitized T cells then in the second step will respond to the antigen after a repeated contact. So after repeated contact with the antigen, CD4 T cells recognize these antigens on antigen-presenting cells, and they release inflammatory lymphokines, which are a type of cytokines. And this, of course, leads to macrophage activation. These activated macrophages then cause phagocytosis of target cells. Now, if we talk about CD8 T cells, they recognize the antigens on somatic cells, and they cause cell-mediated cytotoxicity that directs cell destruction. Now, examples of type 4 hypersensitivity includes contact dermatitis, which occurs due to poison ivy. So if we revise one last time, the mnemonic for hypersensitivity reaction was acid. Now, type 1 reactions are anaphylactic or immediate reactions. Type 2 are cytotoxic or antibody-mediated reactions. Type 3 are immune complex mediated reactions. And type 4 are delayed or T cell mediated reactions. So that's all for this video. I hope now you have a better understanding and you can now differentiate between these 4 types of hypersensitivity reactions. Stay tuned on sqadia.com for more such videos.